Okay, today we're going to have a little look at enzymes. You may remember from last year what enzymes are. They are biological catalysts. That has not changed coming into A level. So biological catalysts, they speed up a reaction. They're all over the place, all over the body, and there's roughly about 40,000 different enzymes in each human cell, and they obviously control different chemical reactions. They speed up the chemical reactions, probably by about 1 million times. So if they weren't there, things would be really slow. Basically, all they're there for is so that these reactions can take place at normal temperature. Because as you increase the temperature, the reaction speeds up. But if we heat our bodies up too much, we'd die. So we need these enzymes in there to catalyze these reactions as quick as possible. Actually discovered in about 1900. Actually discovered in fermenting yeast, and the word enzyme actually means in yeast. So as well as catalyzing all the metabolic reactions, they also act elsewhere in the body. You'll see them in protein pumps, you'll see them in receptors, you'll see them in different motors around the body. So they are really, really important set of molecules. So how do they actually work? So we're going to do this in 3 stages. We're going to look at the sort of biochemistry level, how they actually work, what is happening in there. You may have remember the word active site from last year. We're going to look at a couple of alternative models as well, and then we'll take a look at how they actually do so by lowering the activation energy. So let's have a look at the active site then. So the active site. You may remember we sort of usually draw it as a bit like a Pac-Man. So there's our enzyme, and there is our substrate. So that enzyme is going to break down the substrate, and that will form our products. So 2 models. Let's go up here. 2 models. First model is one called the lock and key hypothesis, and that's essentially as it sounds. So you have your lock, which is the enzyme, and you have your substrate, which is the key. The substrate is the exact shape to fit into the enzyme. So if we look down here, obviously this shape here will fit in there, and you'll get a breakdown into a product. So it's complementary. That word crops up a lot. It's complementary to the substrate. It fits perfectly, and basically the active site, the properties of the active site are actually given by the amino acids around it. So they're dictated by amino acids around it, and depending on the shape of the amino acid depends on the shape of the active site. So the amino acids, they'll actually form weak hydrogen bonds. Okay, they form weak hydrogen bonds, anionic bonds as well. So the active site binds with that substrate only. So that's our first model, the lock and key hypothesis. That's the one you'll have been told at GCSE. The second one is actually called the induced. Go back to that, rub that out. Induced fit model. Induced fit, again, it's as it sounds. So you have your enzyme, let's say it looks a little bit like that, and you have your substrate like that. What will happen is this enzyme will actually change shape slightly to fit the substrate there. It'll change shape slightly. It's flexible. It's not exactly complementary, but as it starts to bind, the active site then changes to fit it more closely, distorts the molecule in the active site, and making it more likely to change into the product. This is the one that has got a little bit more weight behind it. Again, people are not sure exactly how it works, but it's got a little bit more weight behind it. So 5 minutes is up. We'll continue on the next video.